So, do you have any idea about the cake database? What is the cake database? Yeah, anyone?
Ma'am, I have not learned about it.
No problem. So, KEG database, it stands for. Can you see my screen?
Yes.
Yes,
Yeah.
ma'am.
So, KEG that stands for Kyoto Encyclopedia of Genes and Genomes. So, the full form of KEG is what? It's a Kyoto Encyclopedia of Genes and Genomes. So actually, KEG is famous for the pathways. Okay, it is famous for a pathways, the image of the pathway, information of the pathway. But along with the pathway in the KEG, you can say it's a collection of the databases which is dealing with the genomes, biological pathway, disease, drug, and the chemical substances also. So this all information you will get from the cake. But our main focus here is a pathway because we required a pathway many times whenever we are performing a data analysis part. So you should know how to retrieve it correctly from the database. So the KEG database project was initiated in 1995 by the Minoru Canisia professor at the Institute of Chemical Research Kyoto, at the Kyoto University. And according to the developers, KEG is a computer representation of the biological system. Why? Because here you are getting the information of genome, pathways, disease, drugs, chemical substances, and everything. Now, uh, actually, the concept of the cake is realized in the following databases of cake, which are categorized into the system, genomic information, chemical, and the health information. Okay, so if I will show you the homepage of the cake. Hmm. So this is a homepage of the cake where, first of all, it is showing you to ask the, like, this is a search box, and here you are having the cake do. So in the cake too, like you can search the reference cited in the cake, like a PubMed ID, author name, title name, journal. If you wanted to search the reference sites, then the number of references, how many total number of references we are having, which are submitted in the keg. So cake 2, it is showing you the table of contents basically, the information. Out of that, how many pathways we are having, how many bright we are having, how many module we are having, then how many keg orthology we are having, KO, that stands for keg orthologs. Then how many genome information you are having, so how many genomes, genes, glycan, reaction, enzyme, network, variants, and disease. So that everything you will get it here. Then, as I have told you, the KEG, it is categorized into a four categories. One is the system information, genomic information, then chemical information, and health information. So in the system information, the entry means like what sections are coming, like a pathway, bright, module, and R module. So pathway means it is showing you the pathway map. Bright means it is showing you the hierarchies and tables. Module means it is showing you the reaction modules. And these are the KEG R modules, like this modules it will contain. Then genomic information, it contains a KEG orthology, genes, and the sequence data, and the genome data, virus data. So here if you will see like a functional orthologs you will get, then here you will get the genes and protein information, their sequence data also. Then here you are getting the genomic information of a cellular organism and the viruses. Then in the chemical information, it is categorized into four: compound, glycan, reaction, and enzyme. So compound means you will get the metabolites and other small molecules. Then glycan information. Then here you will get the biochemical reaction and the reaction classes. And here you will get the enzyme information also, along with their nomenclature and the sequence data. And at the last, the health information. So it is showing you the network variant, like a disease related network you will get for the human gene. Then you are getting the disease information also and the drug information also. So this kind of information we are getting in the KEGG database. So that table it is provided here. So here you are getting the pathway information if you wanted to search a pathway. So cake pathway is what? It's a collection of manually drawn pathway maps which is representing our knowledge of the molecular interaction, reaction and the relation with the network. So what kind of pathway you are getting here? Metabolism, genetic information, environmental information, cellular process, organism system, human disease, drug development. So related with this, you will get all the pathways. Now, how to search? So basically here, first of all, you have to give the prefix. Means if you are searching for the organism, so every organism is having a particular prefix over here. So that prefix you have to give, the name you have to give, or you can give the ID if you know. So we will see that how to give the prefix and from where you can get it. But at the downside here, they have given the pathway identifiers also. So pathway identifiers means what? Actually, it's a combination of 2 to 4 letter prefix code and the 5 digit number. So if you are having the map as a prefix, so it means it's a manually drawn reference pathway. They drawn manually it. KO means it's a reference pathway which is highlighting the KEGG orthologs. EC means what? It is reference pathways for the metabolic highlighting the EC number, enzyme number. RM means it's a reference metabolic pathway highlighting the reactions. So these pathways are related with what? Like they are related with the orthologs, enzymes, then reactions, so that you will get. And here you will get the numbers. Like if you are getting a 011, so it's a global map. Then 012, it's overview map. 010, it's a chemical structural map. 07 is a drug structure map. And other means like a regular map. And here this is how basically you are getting the, and it is integrated with the module and networks. Like if you are getting M, so it's a module, R means it's a reaction, and N means it's a network. So this is how basically you are getting the prefix over here. So if you will see this, so 011, 011, it's a global map. You can see which is linked with the KEGG orthologs. And M means what? It's a module, and this is a metabolic pathway. So this is how the list of the pathway is given. Maybe if you are searching any one of the pathway, if it is listed over here, you can check it out also. Otherwise, we are having the search box where we can give the entry. Okay. Then here we are having the bright information. So it's a hierarchical classification system for the various biological objects, like which is representing the KEGG objects, like uh, means like a hierarchical information basically you are getting over here. Then here you are getting the modules. So this database is consists of KEGG modules which is identified by M. So module, if you wanted to identify, it is started with the M number and the KEGG reaction module here you will get. So that complete information is provided here. Actually, as our focus is a pathway, we are going on that, but I'm just showing you what kind of information we are getting. So here you are getting the cake orthology information also. Then in this, if you want to search the gene and the gene is related with what other pathways, it is involved in which pathway, so that information you will get it over here. Then here you are having the genome information, virus information, then here you are having the sequence data and the enzyme information. So this is just related with the like right now we are just focusing on a pathway. What kind of pathway we are getting? Metabolic, signaling, and disease pathway. So without wasting a time, if you will just go with the exercise question, you will understand it automatically that how can we get and what information we are getting related with the pathways there. So retrieve the glycolysis pathway for Homo sapien. Okay. So for the glycolysis pathway, you just have to go with the pathway and you can type. Then organism, if you will just click here, right now I don't know what is the code for Homo sapien. Okay. I'm just going to click here on the organism and I'm typing here the human or Homo sapien. So it is automatically giving me the hit. I'm selecting it, click on select, and it is taking the code HSA. Maybe if you want for some another organism, let us consider I want for cow. So cow, you can type, otherwise a Bos taurus, that is a cow, and you have to select it, so it is going to give the code. If you know the code, you can manually type also. If you are not aware, click on the organism, you will get the code. So as we wanted for the Homo sapien, I'm selecting the organism HSA and typing the name glycolysis and click on go. Hmm. Now for this, you are getting a total 40 hits. But all those hits it is showing where the glycolysis keyword is written. Okay, where the glycolysis keyword is written. So here you will get the thumbnail image. Then here you are getting the name of the pathway. You are getting the description of the pathway, like what objects, molecules we are having there. And here we are getting the legends, means like a version numbers of that particular entry. So this is what you are getting over here. Now, here in the names, if you will see that glycolysis is written only here. Other in other entries, there is no glycolysis word. So this is our first pathway, glycolysis or gluconeogenesis. Now, if you will just open this HSA ID, here you will get the complete information of the pathway, like what is the entry ID and what it is. It's what it's a pathway. It's bright or KO means like a cake orthology or a module or a drug. So that information you will get it over here. Then name of the pathway is a glycolysis or gluconeogenesis. Then next is description related with the glycolysis. So what is the description that you will get it over here. Class, it is coming under the metabolism. But what kind of metabolism? Carbohydrate metabolism, right? So glucose is converting into the pyruvate. So it is carbohydrate is metabolized here. Now you are getting the pathway map. So this is what the image you are getting. You can open the image in the new tab. Then here you are getting the different modules. Glycolysis, it is also called as the M2T7 pathway. Then here, 3 carbon compounds pathway also you are getting. Then gluconeogenesis from oxaloacetate to fructose 6P. And another is a pyruvate to the acetyl CoA. So that different modules you are getting here. Then here in this you are getting the network element, the network information you are getting. See that in the network it is showing entry, glycolysis, then definition, class, it's a glycogen metabolism, and in which genes it is involved. So that gene information also you are getting in the network. Then which drugs are like which drugs are useful for it. Maybe there is some change means or you are having a disease which is responsible by the glycolysis, or maybe there is some change in the glycolysis pathway. So which drug we can use there, so that drug is also given. If you want to see the information of uh, glycolysis pathway in the another databases, that link of the databases is also given. GO is nothing but the gene ontology. Organism, that is a Homo sapien. Then here you are getting the gene informations. Okay, so that complete gene information. Then what compounds are involved in the glycolysis. Then here you are getting the reference, author name, and here you are getting the related pathways. What are the related pathways with the glycolysis? That also you are getting. And here you are getting the KO, means the cake orthology ID also. So this is what you will get a complete information regarding to the pathway. Okay. Is it fine?
Mm, yeah, ma'am.
Yes.
Any question?
Hello?
It's not as like vast like NCBI and UCSC. The databases which are seeing right now, these are very small databases. It's just that what is the use, because frequently we require this databases maybe in future, as they are very popular with a for particular entry like a pathway for the protein, for the article. So that's why we are just seeing those databases, because NCBI and UCSC, if you check that, they are very vast databases, right? But this is not like that. It's a very small database, and we are getting the required information from here. Okay.
Ma'am,
Mm,
I have a
yeah.
doubt.
Yes.
Ma'am, could you please repeat the prefix that you have chosen for human? How did you do that?
Okay. So you just have to click here for the organism, and here you can type Homo sapien, human, or whatever your organism is. Click and then select. That's it. It is coming with the prefix HSA. Okay.
Yes, ma'am.
Yeah. Fine. Hmm. So the next question you can try: find the insulin signaling pathway for Homo sapien and list the three genes which are involved in that pathway. Yeah, right, Sonali ma'am. Yeah. What about others?
Ma'am, got it.
Okay.
Yes, ma'am, done.
Yeah. Then sender genes like or okay, fine, no problem. If it's done, then uh, search the gene EGFR provides the official gene symbol, description, and the list out the three organisms in which the gene is involved. So, what is the question? Search the gene EGFR, provide the official gene symbol because sometimes like official gene symbol is different and the gene name which we are searching it can be different. So we have to provide the official gene symbol, the description of the EGFR gene. What is the description of it? And list any three organisms in which the EGFR gene is involved. Can you try this question? Right now, just see because here it is not asking for a pathway, it is asking for a gene. So, where we have to go now?
In the genes.
Okay, yeah, correct. Now, here in the genes, I'm giving the name EGFR and go. So, here we are getting the list. Now, what you have to do is you have to give the official name of EGFR, description of EGFR, and tell me the three organisms where it is involved. You are having total 417 hits.
Ma'am, can you repeat the question?
Yes. Yeah, you can see. Can you see the question? Third question.
Ma'am, in organisms, it is showing humans, chimpanzees, bonobos.
Okay, correct. Then description. Yeah. What about others? Like, are you getting how to perform it?
Yes, ma'am.
Yeah. Okay. So,
Ma'am, can
one
you tell me
single.
description once again?
Yes. That's one simple trick I'm using here. I'm doing the control F and I'm doing the EGFR. Okay. Whenever we are searching the EGFR here, if you will see, the EGFR is highlighted, but here this is along with the RS, along with 1. Here also like EGFR 3, EGFR BP. So there is a no, right now these genes are not having the EGFR, correct? So next here, if you will see this is what the organism first of all, which is just showing the EGFR gene, correct? And what is the description is epidermal growth factor receptor. Okay, so that's a description. Official symbol is the EGFR only. And now we have to see the organism, three organisms where it is involved. So here you are having the HSA. HSA that is Homo sapien. In Homo sapien it is involved. Next, you can see this organism PTR. So right now from the PTR you are not able to understand what organism it is, right? You can just open this in a new tab. And you can see it's a plant troglodytes chimpanzee. That's a second organism. This is PPS. You can see what organism it is. It's bonobo, paniscus. Otherwise, some another organisms are given here. GGO, that's gorilla. NLE, you can check what organism it is. Correct? Are you getting my point?
Yes,
We
ma'am.
can search this from the ortholog table also.
Yeah, you
It's
can
there
try that. Okay, let's try.
because I went there, so I got a big table.
Mm-hmm. So,
It's the same results. So I
but
went in the pathway only from pathway.
means like here.
Yeah,
Oh,
EGFR.
here you type the name
Yeah,
EGFR. Okay.
yeah. Okay.
Hmm. So,
Yeah. Then I click on the entry, first one,
okay. Hmm.
and then just below the diagram there is this ortholog table.
Hmm. Yeah, that's like what a cake orthologs also you can use. Hmm. Yeah, right. You can do it from here also. Okay.
Yeah.
Now identify the disease associated gene in a pathways in cancer. Entry HSA. So I just want to tell you that here by the ID also you can search. I'm just going to copy the ID. Then you can go with the pathway. You can just give the ID over here. Then give the prefix HSA and click on go. So it is showing you the pathways in cancer. Now here I'm just going to be open the pathway image. Also you can open the image, open the in new tab. Yeah, here you can see the cancer pathway you are getting, what genes and all it is having, it is involved. So that complete information you will get. Then once you're opening the pathway, this is some additional information I'm telling because this we have already discussed, right? Then here you can just give the color. You can enter the object as per the line followed by the color specification. Then you can give the. I will show you how to give the color also with some another tool. Then you can just edit the pathways as per your choice. Scale. You want to increase, reduce the scale. Wanted to search the particular ID, you can just search that. Then here you are having the network information. You want to see the ERK signaling cancer. So if you will just click here, you can see it is highlighted, right? All those entries. So this is how you can just maybe on some paper you only want to highlight a specific network. So you can just click here and you can just take a screenshot or you can download the image also. You can go with the download here and then click on the image file. So it is going to download. Organism. For which organism you want, you can just select the organism menu. Okay, you can select it here. Then pathway menu also you can select. See, you if you want to change the pathway type, you can just select from here for you want for Homo sapien or manually drawn pathways or cake orthodox pathways for which animal you want. You just select. You can just select it from here. Maybe I want to see this from the gorilla. You can just select it, and here it is showing you the pathway for the gorilla, right? So that's what you can just do the changes over here only. And if you want to find out the genes in the pathway, you're getting the genes over here.
Ma'am, what was KO?
It's cake orthology. Yeah, I have told you
Yeah,
here, right?
yeah, yeah. Okay.
Hmm, hmm. So here these are the genes which are responsible, which are involved in the pathways in cancer. So this is what basically the uh, information regarding to the images and the what entries and all it is having. But now let us consider there are such kind of pathways over here. I want to highlight a specific genes in or gene in all the pathways. Okay, means let us consider as per the question here, which is given, highlight the EGFR gene in blue color in all the pathways in which it is involved. So what we have to do is we have to highlight the EGFR gene which are involved in whatever the number of pathways, maybe like uh, we want to highlight the EGFR gene in blue color in all the pathways in which it is involved. So for that, in a KEGG, we are having one tool called KEGG Mapper Search and Color Pathway. Okay, you have to go with the KEGG Mapper Search and Color Pathway. Click on the color. Now, for the Homo sapien, I want to highlight the EGFR gene in all the pathways in which it is involved. So what I will do, actually this data we required, it's a tab separated. So that's why it's better to give the input in Excel. Means like we are just going to copy paste it from the Excel. So what we want the EGFR gene because this is a main gene and that I want to highlight with a different color. And I want to give the blue color. I'm not giving the green because when you checked about other genes here in the pathways, they have already highlighted with the green in color. So if you want to change some color, better to give any another color other than green. Okay, so I'm giving a blue color. Let's see if it the genes in which pathway it is involved and whether it is showing the list of pathways first of all or not. So I'm just going to copy this and you have to make sure the color you have to give in a small case only. If you will give blue like B capital or completely blue capital, sometimes it is not going to work. So give in a small case. And here I'm just going to paste it. I'm selecting the Homo sapien for HSA. HSA, that's for Homo sapien. But if you don't want for Homo sapien, you want for some another organism, you have to click on other organism and get organism code. And you can just type the organism, whatever you want, and then select it, just what we did previously. Now I'm selecting the EGFR and sorry, I've pasted this and then click on execute. If you have any file, you can just choose that file here. And here you can see that total 48 pathways in a human where the EGFR gene is involved. Now, this is showing you the ID of the pathway, name of the pathway, organism, and the gene from our list which are involved in the pathway. As of now, we are just giving only one gene. But let us consider you give total 10 genes. Out of that, if it is showing you 8 number, so it means the total 8 genes from our list are involved in that pathway. Okay, so that's a number of pathway. If you will click here, you will get the name also, that which gene it's involved and what is the ID of that. I'm just going to click on that one so that it will be appear as it is. Now, if I will just open the pathway, this which is parathyroid hormone synthesis. Yeah. And here, if you will see where the EGFR gene is there. Yeah, see this EGFR is highlighted here in the blue in color. Is it clear?
Yes, ma'am.
Yes.
Can you try this? You can see any another pathways also if you want. Maybe any random pathway I'm going to open, and it's not like that. You can give the list of multiple list of genes also. Okay. Yeah. See RTK. RTK is another name of EGFR. Yeah, what I'm just telling you is you can use a multiple list. You can see the example they have given actually. See, like ID of the gene they have given pink color, then red color. So this is also the list you can give, but make sure it should be tab separated. Okay. Can you try this?
Done, ma'am.
Okay.
Ma'am, I
What about?
used red color, so I was able to do it.
Yeah, yeah, it's okay. Like color, it's your choice for now because we are just doing it for practice. So actually, why it is why we where this tool will important like whenever we are performing the differential gene expression analysis in RNA-seq or microarray. So some we are having a list of genes, and out of that, some genes are upregulated, which is highly expressing, and some are downregulated, which are suppressing. So in because pathways are not depend upon the expression, right? So one pathway may contain both the genes, upregulated also and the downregulated also. So at that time, if you want to differentiate the upregulated and downregulated genes in the pathways, you can give the different colors. For upregulated, you can give the red color. For downregulated, you can give the blue color. So this is how we can understand that, right? So that's why this particular tool is useful for us.
Ma'am,
Okay.
in the it is saying upload file. So I'm
Mhm.
assuming that we can upload the identifiers along with color specifications in a file format, right?
Yeah, in a file format like
So the
CSV,
file,
TXT,
okay.
but make sure it should be in a tab separated.
Okay, so we cannot upload like Excel directly, right? It has to be TXT, CSV, one of the compatible
No,
one.
no, no. Like entries in the it's a entries in the file. It should be tab separated because here also you are having the tab separated data, right?
Yeah.
Yeah, but generally, when I was checking, it is giving some problem for the Excel file. But when I upload the CSV file, sorry, text file, TSV file, not CSV. Uh, so at that time, yeah, it is generated the result. So
Okay.
better what you can do is because sometimes whenever we are giving input manually by tab separated, it will give you the error. So give the entries in Excel, save it into the text format or TSV format, and then give the input to the KEGG mapper. Okay.
Okay, ma'am.
So this is about the KEGG database.
Ma'am,
Hmm?
ma'am, if suppose we are having 10 genes
Hmm.
and we color code, like we give them a particular color and then we search it, so will it show only those pathway which have that 10 gene, or there might be
No.
some pathways which have 5 genes or 2 genes, that also.
Yes, yes. It will, if it is showing some pathway maybe only having the one gene from the list, that also it will show. Some pathway will have only 5 genes, that also it will display. Okay?
Okay, ma'am.
Yeah. Any another question?
No, ma'am.
No. So this is about the KEGG database. Yeah. Um, Actually, we don't have a session on Saturday, Sunday, but and I in just 10-15 minutes, I'm not able to cover the next pathway. So then, like, because it will give you a problem for the recordings. So we have to start our next topic on Monday only. That's a UniProt database. We are going to start the UniProt database, which is a protein database for the protein functional. It is giving you the protein functional data, also as well as the sequence data. So we'll see that on Monday. If you have any question, you can discuss with me.
Thank you, ma'am.
Yeah, you're welcome.
Ma'am, I wanted to ask, how
Mhm.
will we search upregulated and downregulated in that format? Means the coloring of the upregulated or downregulated in it. Will that will be like we have given EGFR and color, and then we have to give again a new identifier like upregulated or downregulated like that way?
No, no, no. Actually, that you are getting whenever you are performing the RNA-seq based on the specific value. Actually, right now, see, whenever we are performing the RNA sequencing or differential expression analysis, you are getting a value, you are getting a specific column for that called fold change. And in that fold change column, you are having a expression values. So we can apply the filter to those expression values. If it's greater than 1, that's upregulated. If it's less than or equals to -1, that's downregulated. So based on the values, basically we have to apply the filters. And
Ma'am,
once
which
we
value?
huh,
Ma'am, which value?
fold change value, fold
Okay,
change.
so means by this
Okay,
fold change,
fold
the
change.
fold change value from the R class we have done, so we
Yes,
can color it that by that our
yes,
color choice.
yes. We have to filter first based on the fold change value. Then what are the upregulated genes? You can give the red color. What are the downregulated genes? You can give the blue color, and then you can combine it together. Means like in an Excel sheet, all the lists.
Sure,
Yeah,
sure. Thank
that's
you.
how we can know.
Yes.
Hmm. Because if you are searching the EGFR, it can be upregulated at some uh, point and downregulated at some. So we can't mention the identifier there like.
Yes, ma'am. Got it.
Hmm.
Thank you.
Okay. Yes. Okay. If you don't have any question, we can disconnect.
Thank you, ma'am.
Thank you.
Thank you, ma'am.